Hello, everyone, and welcome to today's webinar, Heterologous Protein Engineering and Characterization of Native Regulatory Elements in E. coli using Massively Parallel CRISPR Editing. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Tyson Shepherd, Microbial Engineering Applications Manager at Inscripta. Dr. Shepherd, you may now begin your presentation.
Hi, thanks so much for that introduction, and thanks to all of you for joining me today for this upcoming webinar. In this webinar, I'll be taking you through a recent application that I've been developing while at Inscripta. So this application is leveraging the Inscripta Onyx platform to be able to make protein modifications and improvements to the functions of proteins that have been stably cloned into the genome of E. coli in this case, and then being able to create an engineered variant of that protein and to be able to modify expression. So as mentioned, I'm a senior scientist in the applications development group at Inscripta. I've been here for about a year, and so yeah, let's jump in and webinar. So I think we all know that, of course, evolution is a driving force behind being able to diversify new activities, which is Saccharomyces cerevisiae, where you can actually design your mutations specifically to these to the genome, either customized with heterologous proteins, as you'll see later, or with the native genome directly. You then ultimately will get to order those libraries, and then in a few weeks you'll receive a consumable kit that you load to the instrument, and the instrument works in an automated way where it will then manufacture a pool of cells that in aggregate contain the designs that you've designed on that portal. That engineered cell is then taken forward to do all sorts of fascinating phenotyping. That's where you can stretch your legs on what you can do with hundreds to tens of thousands of different variants in a library pool. And after your particular phenotyping assays, you can take that back to the Inscripta resolver to be able to connect back the phenotype to the genotype. And that's really the workflow that we're going to walk through today with the heterologous protein expression. So the Inscripta offering here, the best way to kind of think about it is previously, if you wanted to design a number of variants, what you would typically do is separate those out into a microtiter plates, and then do your CRISPR gene editing or genome editing on those individual wells within that and grow those up in a singulated fashion. You'd introduce the single guide RNA to make the cuts, and then, of course, the donor DNA to make the repair happen. We're kind of changing the way to think about that because we have a barcoding system that allows you to be able to track a cut and donor DNA delivery to an individual cell. And so ultimately, we have in aggregate the cell itself being behaving as the partition instead of the well on the microtiter plate. That allows you to be able to realistically track from hundreds to tens of thousands of different edits in an individual library to be able to screen them or select from that pool of all of them. So as I mentioned, the number of edits and the type of edits that you can make is, you can think of substitutions or insertions of up to 63 base pairs, or deletions up to the same size in either non-coding regions, which lie outside of the coding sequences. And here you might think of targeting transcription factor binding sites or promoter sequences or ribosome binding sites, for example. We also have the capabilities in the designer to target the coding regions. And that really allows you to dive into being able to do saturation mutagenesis and gene knockouts or truncations, for example. And so it's really up to your imagination at that point of being able to port over new biology to engineer it to your desire. So just to give you an overview of the system is with the initial hosts on the initial offering, the hosts that we're supporting are E. coli and Saccharomyces cerevisiae. We have a standard set of strains which are based on, for E. coli, MG1655, and for Saccharomyces cerevisiae, S2C, and SNPK. The genome targets are both coding and non-coding regions in that genome, and then also heterologous sequences that are introduced into the genome, so a custom type genome. The design library size for E. coli is from 100 designs up to 10,000 designs, and for Saccharomyces cerevisiae it's from 100 designs up to 6,000 designs. And to keep in mind about the actual designs that can be made is that you will have the capability of making one edit per cell, but in aggregate it will contain all of the designs that you've pushed to it. It can be insertions, deletions, or substitutions from one base pair up to 63 base pairs. And on a typical library composition, on a typical run, you would see greater than 90% of the designs covered through sequencing, and with an edited cell population of between 15 and 75% of your cells being edited. The time to carry out this particular task is, it takes approximately 3 weeks to be able to receive the consumables, and an on-instrument run is about 2 to 4 days, and the hands-on time is pretty much less than an hour to be able to load the consumables and press go. So that's been a tremendous savings, and something that I'll highlight through this webinar is the time savings for doing protein engineering. So I show here now that you have in your mind what the Inscripta Onyx platform has as capabilities, I show here a number of different possible application areas that we've been interested in, but I also think that generally that will be impacted across the community. So I reference here in strain engineering is that this opens new doors in metabolic engineering and strain optimization for allowance of expression of toxic cells, toxic proteins, or other small molecules that would inhibit growth. Also engineering heterologous pathways. So as larger and larger DNA becomes engineered within the genome, that becomes more accessible to engineering with the Onyx system. We also generally have interest and seen interest in antibiotic resistance and applications to being able to develop and understand routes of new antibiotic resistance. There's, of course, questions around evolution and achieving new enzymatic functions by modifying enzymes based on evolutionary characteristics. And then separately, there's, really rich research that's going on in regulatory networks and being able to understand the actual causal interactions that take place within a cell signaling cascade. All of these are accessible now with the Inscripta Onyx platform. And so it does seed the question as to how would you use this type of platform if you had the capability of making tens of thousands of different variants within a library pool to test hypotheses. So we generally think in terms of breaking down the workflow into two different possible workflows. One is a pooled selection type approach where we take that edited cell population and we want to interrogate the entire population and see its behavior. So there we might be looking for enrichment in a small molecule growth inhibitors. And so I encourage you to go out and look at other webinars that we've done previously that have talked about this pool-based approach and the selection mechanism and the route that you would take through phenotyping on selections. But a separate route is equally as interesting as this singulation of colonies and looking at a screener's workflow. So in that case, you would take your edited cell population and separate them out into isolates and then be able to test those isolates for new or novel functions. And that's the workflow that I'm going to highlight for us in today's webinar. So in that route, what you would typically do, you're not selecting for a population in this case necessarily. What you might be doing is looking for a particular feature that then you would want to drive back into sequencing to be able to connect that up with the genotype that you've generated. And that allows you to be able to connect that phenotype back to a genotype where you can really stretch your legs on learning new things from the biology. I'm going to highlight two application areas that use that isolate workflow. The first application areas is small molecule production, so testing variants for being able to improve overall production. In this case of lysine. So of course, with E. coli, lysine of interest is one of a number of different small molecules that can be generated in E. coli for industrial use. But this is really a vehicle of showing the power of the Onyx platform to be able to really expand the amounts of biosynthesis that can take place when you properly engineer a chassis for production of that molecule of interest. So there's really three different scopes that you can look at in editing. Of course, it is at the protein level. Each of the biosynthesis steps realistically occurs at the active sites of the enzymes that carry out the synthesis reactions. So that's where the magic of saturation mutagenesis in those active sites can take place. The next level up are the pathways that are responsible. So it's not just going to be about the individual pathway of the biosynthesis, the proteins that are interacting, the active sites of each of those biosynthesis proteins, but also the pathways that feed into that biosynthesis. So, for example, those which would otherwise go off into methionine and threonine synthesis, those also are valuable targets because they allow for the intermediates to not be diverted away from the production of the small molecule. And then to think more expansively is what can you affect on a genome to also be able to improve production? And these are often things where you don't really even know what the connection is at the start. It's exploratory to be able to identify different proteins across the entire genome that are responsible for improving the overall production. These might have to do with toxicity of the small molecule. It might to do with being able to get energy supply to the biosynthesis pathway, etc., etc. So really, it's at that point is at the horizon of being able to modify the chassis to be able to be perfect for the small molecule that you're producing. So for lysine, what we did was a screen of what ended up being 18,000 different isolates that we screened. So this relied heavily on a rapid-fire mass spec to be able to identify the production across all of these different screened isolates. So the way that we got to build up such a massive library to test was we established a knockout library, a genome-wide knockout library. And each of those knockouts took place in two different ways. It's nearby to the start codon, and then just a little bit further in, but still stopping early, terminating early in every protein in the entire E. coli genome. Secondly, on the right side of the left figure is the promoter libraries. So I've talked about these in previous webinars. This is a pretty fascinating concept of putting a constitutive promoter in front of every coding sequence in the entire genome. And so we built a ladder of weak to strong promoters to be able to test how much of an impact those promoters would have across the entire genome. And then in the center point are all the edits that were made targeting to the specific pathways. And so that's really a lion's share of the edits for this library. 174,000 different designs were made that were targeting the actual pathway. And here it was really targeting pretty broadly all of the genes that were involved in that lysine biosynthesis pathway and the diverting pathways. And so then, as I mentioned, we had a total of being able to synthesize these. We ended up totaling 24 different libraries that were generated, and that in total, in aggregate, the entire pool size that we were testing was over 200,000 designs. So we really needed that high throughput type system. So in this case, we relied heavily on mass spec. For pathway engineering, that central library that had that lion's share of edits, there's 19 total lysine biosynthesis pathway genes that were targeted. In addition to that, there's 20 transcription factors that are responsible for interacting with and ensuring expression of these pathway genes. And so all of these were targets then for site saturation, mutagenesis, and being able to mutagenize the sites. It turns out that we were able to, by that screening campaign, identify 138 unique hits that improved the production on the first round of testing. So the improvements that we saw from that mass spec were from anywhere from twofold up to 1,000-fold over the wild type. So we already saw a substantial improvement even by one round of editing on the Onyx workflow. So it really was that capability of being able to have access to all of these different variants in a very rapid way. So all of these can be generated on the Inscripta designer software, and then you would receive these various libraries and be able to load them to the instrument, and literally within weeks of the initial hypothesis that you have, you're to the point where you have a pool of cells that have all of these different possibilities in it, and then it's just left up to your screening campaign to be able to dive into that cell pool and isolate out the best, the winning variants in that population. So we really have that kind of rapid turnaround that's unprecedented in our fields. And that's one of the things that makes me so excited about this platform. So certainly, like I have a background in structural biology, and so I think of these often in structural details. And one particular protein that we really saw a number of interesting hits on is this DAPA protein. So this is a central or key enzyme in the lysine biosynthesis pathway. It has this feedback inhibition by the lysine production, and so it was hit hard with this library amongst other enzymes by a site saturation Unigenesis Library across the enzyme. And one thing that's of note about this enzyme, the DAPA enzyme, is that it's known in literature to actually have a variant, glutamic acid 84 to threonine, that is known to improve production. And we actually ended up capturing that hit in our screening campaign, which we're always excited about finding these literature relevance captures because it's quite a nice thing to be able to connect that back with the literature findings. And so we saw secondary to this mutation is that by putting a constitutive strong promoter in front of the DAPA gene, we're able to further enhance that production of lysine. So those were a couple of hits that we pulled out. So we as you can see from this plot, the fold improvement over the wild type in just the first round of editing. So this would take, like, you're thinking in terms of like 1 to 1.5 months of the initial idea to get to the point where you've got this 600, 900 fold improvement over the wild type. So there's a couple of examples of that occurs in the first round. That's that literature example of the DAPA with glutamic acid modification. But then there's also this constitutive promoter that was put in front of the DAPA, and there we saw almost 1,000-fold improvement by putting the constitutive promoter in. So you have to be wondering, what happens when we start stacking these modifications on top of each other? This is what we saw with the second round of editing. So we took our winner from the first round of editing, a DAPA with a high-strength promoter, and built on top of it yet another round of editing. So this would be an additional, if you had the reagents ordered, or even if you're stacking it on top, you're looking at 2 months, 3 months to get from original hypothesis, now getting 10,000-fold improvement of production of the small molecule. So combined in aggregate, when putting that mutation together, we see this huge increase of so that's kind of the winner to take into the next rounds, the round 3 editing. So we take the winner that has that mutation, and we push forward with a genome-wide screen of hits. And this happens in the pathway itself. We captured one where it had another constitutive promoter, the J23-100, in front of this GLMD protein. That further got us to a grand total of 14,000-fold improvements over the course of several months from start to having this enormous E. coli that could produce substantial amounts of the lysine. So that was an exciting story, and again, I want to emphasize that that's a vehicle for or a layout of how it could affect your research. So if you're interested, if you have interest in small molecule production, imagine what you could do with harnessing this technology to be able to build a chassis around this specific small molecule that you're interested in. So I want to talk about now like the core of the talk, which is the heterologous protein engineering. So in this case, this is about protein optimization using the Onyx system. So we just got done talking about how we could manage to see, massive improvements of production using the Onyx system to be able to capture and modify a pathway for production. But we also want to stretch our legs on thinking about what if we want to put in a heterologous pathway, or in this case, produce a heterologous protein. So the protein of choice here is a fairly simplistic model system. It's a GFP. And what I want you to think about is the aspects, once again, of how this actually applies to your own research. When I talk about GFP here, I'm laying out what can be done with multiple different proteins, be it for industrial use cases or therapeutic designs, is generally applicable type of workflow, and that's why I thought it was so interesting to get it out to others. So the workflow for the heterologous protein engineering starts from, of course, generating and doing a QC step on the initial strain. So that initial strain builds. And then that gets loaded up, the final GenBank, the GenBank that includes that heterologous protein gets loaded up into the Inscripta designer portal, which then is able to parse out the GenBank file and allows you to be able to begin making edits on the protein of interest or on the entire genome that the protein is in. And so that's how you come to your design libraries, where that's that Inscripta designer where you get to interact with your genome. You take those design libraries and you get a pool. You ultimately get the consumables that get loaded to the instrument. The instrument gives you a pool of cells that in aggregate contain all those designs, and then it's off to the races with phenotyping. So in this case, I relied heavily on the Toucan plate reader for being able to look at fluorescence. You take those back to sequencing, be it with sequencing the library or sequencing the individual isolates, and then those get taken into the Inscripta resolver platform, which can process out that data and allows you to go back, just like we did with the lysine production, allows you to go back and make new designs now with your new high-performing protein or enzyme of interest. So the way that I built this was not entirely different than, of course, what's been reported in literature to co-opt the CRISPR-based system and combine that with the lambda-red recombination to be able to insert gene-length DNAs. In this case, I used the MAD7. I used the Inscripta's almost like an open source CRISPR enzyme, because you have access to it in a novel kind of, we provide the sequence for it, we provide readily available for research use cases. And here I applied that system to be able to build the genomic, build the pathway into the genome. And so this is, as I mentioned, super folder GFP. I chose that for the high fluorescence. I also put in front of the SFGFP the J23102 constitutive high-strength promoter. It's not the highest strength promoter, but it's certainly a strong promoter. I built this, the double-stranded DNA template, the PCR template, with 55 nucleotides of flanking homology sequence around the cut site. The PAM site was chosen from the MAD7 typical sequence. The CRISPR RNA sequence is provided. So I kind of custom built the particular cut site to allow for integration of the SFGFP. The GFP sequence itself is based on the superfolder GFP. I modified the gene sequence itself to be slightly AT-rich, so it ended up with 45% GC content. And then I also removed chi sites and common restriction enzyme sites, as well as ensured that there would be plenty of Mas7 PM sites distributed across the GFP sequence, and then all that was terminated with the T0 terminator. So this is one standard approach, this CRISPR-based approach. Certainly there's other amenable options for building out your heterologous protein, including using antibiotic markers that can be looped out or left in, or oxytrophic markers, especially, of course, widely used in use for building in pathways. So then the next question is, where do you get the protein optimization variants from? And here, you can go about this any number of ways of being able to look into a whole catalog of different variants. So you can digest those from those insertion solutions. The SNPs are what you're looking for. You can dive into regulatory data and databases that contain that regulatory information. You can look at evolutionary trees and strain variants, so comparing, say, different strains of E. coli and looking for SNPs or insertions and deletions between them to be able to pull out the feature that you want to have. You can do that down to the enzyme scale, so looking at evolutionary variants of enzymes or strain variants of specific enzymes of interest. You can also use structure-guided approach to generating variants. So you can look at the active sites and be able to identify residues important for in that active sites and be able to do a variant library with those. Another good way is to look at published enzyme or protein variants. And so that's kind of the approach here. It's kind of a combination of several, but I leveraged FPBase, which is a database of fluorescent protein variants, and it's got a fantastic catalog of all sorts of great information about fluorescence, excitation and emission data, and even more like quantum yields and so forth. So it's this rich data set. And what I did was I took an alignment of that superfolder GFP against many other GFPs in the same clades for identifying hotspots of mutations that cause some difference in the spectral characteristics. And if the mutation shows up in multiple instances, then I call that a hotspot, and that then gets moved into a saturation mutagenesis library. So this is a pretty targeted library against these hotspots. But it's saturation mutagenesis, so it's all 19 other possible amino acids against each of these evolutionary or variant hotspots across the protein. And so in aggregate, I had 723 designs in total that then I took forwards through the entire Onyx workflow with those 723 designs. So I ran that through the Onyx workflow, uploaded that data using an identification analysis on the Inscripta resolver, and that showed that I had about 66% editing in my pool. So in this case, we're calling that an edit, such that approximately when pulling out a cell, there's like a 66% chance that that cell is going to contain an edit in this case. So that's not saying per design. That is saying that in aggregate there's 66% edited cells in your population. And then we use other metrics to be able to identify how likely your design is. We have a rich data set that plays in through the Inscripta that you can identify through the Inscripta resolver platform for being able to identify how many cells to screen, etc., based on the actual metrics that we get from the data. So we were very happy with this. It turned out that the metrics looked extremely good for this library, very much ready to carry forward. So all of that happened. That design step took a couple of days coming out of FPBase. pushed into the Inscripta designer, which was, within a few minutes you get your designs back that meet your specifications. And then a few weeks later, I got my consumables, and all of this occurred very rapidly. Within 2 to 3 weeks, I was off and running on phenotyping. So right away when plating out for CFUs, I could see a wide variety of different colored colonies on the plates. So it was, I already had a notion this was going to be a rich data set. I was quite excited about that. I picked colonies, attempting to be somewhat agnostic on the color of the colony when picking, and I grew those up over the course of 24 hours, washed them in minimal media, and then diluted them down in minimal media to do a fluorescence screen on. So that was in a TCAM plate reader. I chose 192 colonies, and then I screened each of the 192 colonies at 3 different excitation and emission wavelength pairs. So I based these pairs on blue fluorescent protein, excitation and emission peaks, fluorescence, GFP, peak excitation emission, and YFP, peak excitation emission. So I was able to rapidly screen using this 3 different hypothetical fluorescence peaks to be able to pull out if there was any variation in the actual spectral characteristics. Any of those that were different from normal fluorescence, I then did a full spectral scan on, a full excitation emissions scan on. And what I found was that there was this rather fantastic distribution of fluorescences. So there were, what's plotted here is the fluorescence intensity. On the y-axis is when excited at 510 nanometers, so the YFP excitation. And on the x-axis is the GFP excitation emission, plotted the intensity of fluorescence. So the wild-type superfolder GFPs all kind of clustered around, as you might expect. And a number of the colonies had totally disrupted fluorescence in the GFP and YFP channels. And some had disrupted GFP fluorescence, but substantial YFP fluorescence. And so that indicates that the excitation emission wavelengths at peak have probably shifted. And there was one in particular that really caught my eye. It was pretty far out in the GFP channel. It's about 1.5 times more bright than the wild type superfolder GFP. And I should note that all of these are adjusted on the OD600's absorptions. So they have been adjusted for the amount of cell content. So that really far out spot I highlighted here with an exclamation point. That was a great finding, because I was pretty excited that we found this higher intensity variance. So as I mentioned, the Inscripta resolver makes it trivial to be able to take all of that content that you just gathered from your phenotype and go back to the genotype. So through that, through the sequencing pipeline, I took that to, I also did Sanger sequencing just for the excitement of it, but I took all of those colonies back through Illumina sequencing. I loaded the FASTQs up to the Inscripta resolver and was able to get the identification of every one of these variants and be able to connect back from phenotype to genotype in a remarkably easy way. And a couple of cool variants showed up. So it was the improved intensity. That was very exciting to see, and that turns out to be in superfolder GFP. It's known that this is one of the critical residues is the phenylalanine that's at position 145. And it turns out that the mutation that I pulled out with the improved intensity was a mutation of that phenylalanine 145 to methionine. Showed this improvement of intensity. I also pulled out a number of those that had disrupted fluorescence. It turns out that when I checked on the blue fluorescent protein channel, that was excitation 380 and emission of 445, it turns out that a number of those colonies, while disrupted in the green fluorescent protein channel, were actually quite active in the blue fluorescent, with blue fluorescence. And so taking some of those colonies back to sequencing, several of those colonies identified as the tyrosine 66 to histidine. This is a common and well-known mutation for generating a blue fluorescent protein. So there's that literature, that kind of almost just a really good feeling when you've connected back to literature a known variant. So that ended up with 375 excitation and 440 emission peaks with that single point mutation. So now that we have this improved intensity F145 to M mutation, I wanted to carry that forward for an additional round of editing. Not dissimilar from that story that I was telling you about the lysine production. And on the second round, I picked up a whole other set of variants that were interesting. So a few seemed to have a moderate increase in fluorescence, but there were a lot that clustered around the wild type, now wild type F145 to M fluorescence. There were the pool that got disrupted, but then there were a couple of colonies that really spiked off the charts on the YFP. And one of those in particular had great fluorescence, so that's a novel YFP variant based on the F145 to M that was carried forward. So it shifted from the 480 nanometer excitation to 510 nanometer emission wavelength that shifted to 508 excitation and 522 emission. So it really turned out to be this nice, bright, yellow fluorescent protein. And all of this was able to, like I mentioned, all of this was able to be carried forwards in, a matter of weeks from the start of this. So separately from this, I had an interest in whether or not you could actually screen tuning the GFP expression itself. So still looking at the locus, if we were to swap out or substitute in, instead of that J23102 promoter, we substitute in 50 base pairs that are in front of every transcriptional start site that was annotated in the EcoPsych database. So we swap out all of the, from the J23102 constitutive promoter with every promoter in the entire E. coli genome. And separately make the library where we swap out the Shine-Dalgarno sequence that's built into the GFP locus, and swap out the 20 base pairs prior to every coding sequence. So ideally capturing, or hopefully capturing, every ribosome binding site in the entire E. coli genome as well. So both of those together are 4,000 plexity. So in aggregate, you get 8,000 plexity library. And here, again, by screening individual colonies, you can imagine ways that this can be made to be very high throughput with fluorescence activated sorting. But just screening out individual colonies, I was already able to pick out some interesting things where the control value, none of the native promoters or ribosome binding sites were able to hit that control amount. Like, we've really chosen the synthetic system that should have extremely high expression, and it's living up to its expectation. But what's fascinating is that we get to catalog a number of different promoters and ribosome binding site strengths by looking across the entire library of data. And we can organize those out into what's the highly expressing promoters, medium tier expressing promoters and ribosome binding sites, and really quite low or inactive. Now this could be like annotation issues, or it could be feedback mechanism issues. So there's probably multiple theses worth of data that's contained within this type of library. This is the type of thing that you can have in the palm of your hand once you get that cell pool ready to go. So I kind of broke out these high, medium, and low tiers of promoters and ribosome binding site strengths. And really we start to see some kind of interesting features to it, is that there's a number of these that were pulled out that were chaperones and that are involved in core metabolism of E. coli. There's even a few uncharacterized proteins that find their way into the mix. And so certainly we see this this broad spectrum of ribosome binding sites and promoter strengths, and I'm still quite excited to dive a lot deeper into this particular library. Just keenly interested in seeing how fluorescent sorting might give a whole population of information about all of the different promoters and ribosome binding sites that would be accessible in such a campaign. But I'm just giving you here, like, the workflow where I'm able to pull out this interesting information that can really identify how these promoters are functioning and using a fluorescent screen. So really the objective here is to show you that the creation of libraries of cells with greater than thousands of diverse phenotypes has never been easier. That Inscripta Designer is just an elegant solution to being able to interact with your genome to generate novel variants, and that this is now like being able to do site-directed mutagenesis at the genome scale, to being able to make modifications to protein to be able to carry out saturation mutagenesis on a protein on a genome with precision engineering. Just has never been possible before like this. And the other thing that I wanted to highlight is that this was done over the course of several weeks. So the workflow here is just as simple, if not simpler, than doing a plasmid-based approach. And the key outcomes that I get from it is that this protein is stable on the genome. I know exactly what's in the genome. I know exactly what's the copy number, the tunability of it, and it gives me a whole new aspect for being able to do that precision engineering of the protein. I kind of throw it to you now. So at the scale of 6,000 to 10,000 different edits per library of trackable designs, with 15 to 75% edited cells in a population, all within a benchtop platform that has integrated software, is that what could you do with this, the power of this instrument? If you're interested in small molecule production, if you're interested in pathway optimization of protein production, and previously I've given webinars about applications to laboratory evolution and raising tolerance to small molecules. The application areas are endless, and so I'm really excited to see what you'll be doing with this powerful technology. So thank you very much again for joining me, and I look forward to answering any questions that you have.
All right, thank you, Dr. Shepherd, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question is, what are the benefits of doing on-genome modifications rather than the plasmid-based optimizations of the same type?
Thanks. That's a great question. So the benefits kind of weigh in mainly on what your current workflow is, but for a lot of purposes, historically plasmids have been used for protein optimization and making site-directed mutagenesis because that was the available technology. But often those findings would then have to go through subsequent transference to the genome when scaling up for production, for example. What the Onyx platform really allows us to do is to bring all of the technology that comes from site-directed mutagenesis or being able to access all of the landscape of amino acid changes and so forth, but directly on the genome. What that translates to is a very rapid way of being able to get consistent, reliable production off of the genome, and any changes that are made to the genome are directly available then for subsequent scale up rather than having to go through an additional 6 months to a year of technology transfer as the mutations that were identified are transferred back to the genomic expression. So there's a number of achievable, powerful methods of editing on the genome that are now available through the Onyx platform.
All right, thank you, Dr. Shepherd. The next question, what type of optimizations were used for the gene insertion?
Yeah. So when we've been doing protein production applications, and then I also encourage, we have some webinars out about optimization of an enzyme CBH1, and in both cases, when putting them into the genome, the first thing we think about is where to actually place them in the genome and the workflow to use. So we were using a MAD7 approach to inserting into the genome. For the actual sequence optimizations, we think about what promoter to use, and is that promoter sequence orthogonal to the genomic DNA. We think about, of course, the ribosome binding sites, and in this case, as you saw, we kind of had a pretty high expression of GFP. That's usually a good place to start for protein production. We do codon optimization, and with a little bit of an extra spin on it is that we sprinkle in kind of MAD7 PAM sites throughout the genome, so that PAM sites for MAD7 are AAA, and so when available, we kind of put those PAM sites into the codon optimized gene as well, such that it then becomes allowable to access any site along the gene for editing. And then the final thing, of course, is that we think about is the terminator sequence, and also what kind of flanking sequences that we might incorporate to be able to excise the gene later, etc. So these are the type of things that we think about when we're integrating a gene into the genome to make it fully accessible for the Onyx platform.
All right, thank you again, Dr. Shepherd. Our next question. All the promoters and RBSs were below the synthetic sequences used. Can you comment on this?
Yeah, absolutely. So I had put all I'd made a library off the Onyx that used all the promoter sequences and all the ribosome binding sites that are found in E. coli that then were put in front of and replaced the synthetic J23 series promoter and the maximum RBS. So the J23 series promoter, it wasn't the the highest strength, but it was just off the highest strength. So if anything, that those results definitely clearly indicate that those J23 series promoters, the high expression ones are very high constitutive expression. The ribosome binding site is indeed, pretty high. Now, one thing that I did on that particular application so far was a very just I had isolated and looked at just 192 colonies, and so then when we think about the plexity of that library, the plexity itself, it ended up being about 8,000. A size of 8,000 different designs that went into that library. So I was screening at just a very low depth of screening on a plate-based format. So the promoters that I accessed at that time ended up being less than the high strength promoter. I thought that was rather interesting too. Is a scenario where even with that low depth screening, there were a few promoters that did kind of rise up near to the J series promoter. So even at that low screening depth, there was some interesting details. I think that particular application, once we do a cell sorting type workflow, we might be able to really identify if that holds true for all of the promoters across the E. coli, or is it constrained to just because only a few were tested? So thanks, that's a good question.
All right, thank you, Dr. Shepherd. Our next question here. These optimizations were in fairly low throughput. Do you have any thoughts and recommendations for increasing the throughput to test more variants?
Yeah, definitely. So just as I touched on there, is it was plate-based screen is already fairly good for shallow sampling for being able to obtain those which are trending upwards, but it's really interesting once you're able to use a higher throughput screening workflow, or especially a selectors workflow, where you can really kind of dive deeper with your library. So one of those methods, which isn't shown here, is like cell sorting. So that would be available on a fluorescence based approach. So with cell sorting, you can kind of sort based on the fluorescence. And so in the application that was shown here, where with the native promoters and ribosome binding sites, a cell sorting experiment would allow for binning out those fluorescences which have high and low fluorescence to be able to look at essentially the entire population. So that's always great to think about when using these library based approaches, is what can you do in your workflow to be able to kind of take advantage of all the designs that are present in the 100 to 10,000 different designs that would be available for you to screen on.
Thank you, Dr. Shepherd. Looks like we have time for one more question. Did you test the impact of other genes on the production of this protein?
Yeah, so that's, if I understand correctly, I think that's an interesting question. That's getting at the concept of, can you go genome wide instead of just looking at the gene. So here, this application, I kind of focused down on what you can do on the gene once it's on the genome. But that's certainly very interesting, is could we do like a genome-wide promoter or genome-wide knockout series to actually improve the overall expression of the protein? That for sure is addressed a little bit more with the other app note that I mentioned in the CBH1 enzyme protein production optimization, where we did go genome-wide. Here I have ambitions to take this to genome-wide, but it just became a bit of an interesting little side project to see what improvements I could make to GFP itself. So you can suspect that as you make improvements to the genome, there's going to be many different chaperones that can be upregulated, or proteases that can be downregulated or knocked out, that we could really boost the actual protein expression. And that's something that we're obviously very interested in trying to develop those type of strains with anyone. So thank you.
Thank you again, Dr. Shepherd, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.